Hey guys, it's Miss C. In this video, we're going to get into the endomembrane system. Oh boy, everyone's favorite. No, I'm just kidding. It's actually a pretty cool little system. Why? I want to start by explaining why is it called the endomembrane system and what makes it considered a system. Now, endo means internal. In this case, the endomembrane system, why it's called that, is it is a bunch of organelles that work together to meet one common goal. And in that case, this common goal is synthesizing lipids and proteins, modifying it, and then shipping them off to their appropriate destination. So I'm going to go ahead and write that down on the left. All right. So I went ahead and wrote all that out for you so you have it if you want to copy it down or whatever. What's included now, I said it's a series of organelles. Don't get confused and think that it is all the organelles there are, because it's not. It's actually a really specific sequence of organelles that, like I said, kind of work together to make and ship proteins and lipids and sometimes some carbohydrates to their appropriate place. And it all starts in the nucleus, and I'm going to get into all these specifically. Then it moves into the ER, to the Golgi, and then into secretory vesicles. And sometimes those vesicles are going to wind up outside the cell in they fuse with the plasma membrane, or sometimes they can wind up in these digestive organelles called lysosomes. So sometimes they will take one of two routes. They can either take whatever their contents and dump it outside the cell, fuse with the membrane, or they'll actually wind up inside of these lysosomes. They'll fuse with those, and those will digest them. So I'm going to write that. All right. So let's see if we can make some sense of this endomembrane system. Starting with step numero uno, the nucleus, right? Everybody always thinks of the nucleus as the brain of the cell, probably because that's where all the DNA is located. So first things first, the DNA is located in the very center of our nucleus here, which we call the nucleolus. So a lot of times the DNA is going to be really, really condensed and packed in the nucleolus. And one thing I kind of want to mention right now is because the DNA is located in the nucleus, it makes sense that this is the site of transcription. And I know we haven't talked about that just quite yet, and there will be a whole another video dedicated to just transcription. But I think it's worth noting, just to make some connections, that transcription occurs in the nucleus, and that is the process of going from DNA, which makes sense that this happens in the nucleus because that's kind of like where the DNA lives. It doesn't get to leave. It's kind of held hostage in there. It doesn't really get to go anywhere else. But what does get to leave the nucleus is something called RNA, which is basically a copy of DNA that we don't care about as much. It can be shipped out into the cytosol or to the rough ER where the ribosomes are, and we use it to translate into protein, and then we can destroy it and nothing bad happens, right? You destroy your DNA, and that's going to lead to death and destruction, which nobody really wants to have happen. So moving on from that, I just like to pepper that in here because that's something that we will get back to later on. And I definitely want to make sure that you're making connections, that transcription occurs in the nucleus because that's where the DNA is. So we have this part of the nucleus called the nuclear envelope. And what's an envelope? It's something that has other things inside of it. So, you put a letter inside an envelope and you ship it off to wherever. Same concept. It is basically the membrane that holds everything we consider inside the nucleus. So making a little note here, there's a couple things you should probably know about the nuclear envelope. it's not completely airtight sealed. The nuclear envelope has things called pores, where the RNA will actually, when it's synthesized in here, it'll ship out of the nuclear pores and kind of get shot like a t-shirt out of a t-shirt cannon. It's kind of what I like to think happens. I don't really think it's that cool, but whatever. It gets shipped out of the pores. And then also, you can't really tell in the picture that much, but the nuclear envelope actually it has a double membrane, so it's like I always say it's like a double wrapped Chipotle burrito where you got one tortilla and then just another tortilla wrapped right around the outside of it. So, lastly, it's not even labeled in this picture, which kind of sucks, is something called the nuclear lamina. And I think, well, lamina kind of sounds like laminate, and that's what you're seeing. There's these fibers that are located on the inside of the nuclear envelope, and you can kind of think about them, they kind of line the edges, and it's just for structure. It's to keep it nice and round so it's not getting squished or really screwed up. And that's pretty much the gist of the nucleus. It really is just where the DNA is located, and its role in the whole endomembrane system thing, tying it back, is it's actually physically attached to your ER. So you can see, whoop, there's a little bridge. They're actually fused together, and because this is where transcription occurs, DNA to RNA, that's important in the sense that RNA will eventually be translated into a protein. So moving on to the next organelle in the endomembrane system is the endoplasmic reticulum, which is like way long and fancy. So most of the time we abbreviate that to ER. There's two different types, and I'm going to try to keep it simple. You have smooth and you have rough ER. Rough ER is called rough ER because it's studded with these little ribosomes. So you can see right here, there's a little arrow pointing to the ribosomes, and if you know what ribosomes do, it makes sense that the rough ER synthesizes certain proteins, especially ones that are destined to leave the cell. Secretory proteins that will be secreted out, and they'll be packaged and shipped in the Golgi and all that. So right now, the rough ER is where secretory proteins are synthesized. So that's where they're actually made. And it is rough because it is studded with ribosomes. I like to think of, it's studded with ribosomes, so it's kind of like braille looking. So I think rough, and also rough and ribosomes both start with R, so maybe that can kind of help you remember the difference between rough and smooth. So think rough, ribosomes make proteins, so that it makes sense. Rough ER makes proteins too. The smooth has a couple functions. Smooth ER can synthesize lipids and sometimes some carbohydrates, and it also is in charge of detoxifying. All right, so I think that it makes sense that your liver cells actually have a decently high concentration of smooth ER because the liver, its whole job is that it detoxifies poisons. Don't know why you're drinking those, whatever. Nobody's here is asking any questions, but it does help detoxify. So what's interesting is that your smooth ER actually has the ability to double its size in the span of a day or two, which is why people develop alcohol tolerances. You drink a couple beers, and then the smooth ER in your liver cells actually expands. So the next day you drink 2 beers, you might not feel the same effects because your body literally is better at processing the poison that you're drinking, which I always thought was interesting, and a reasoning behind the tolerance that I think we're kind of familiar with. So those are the real roles of the endoplasmic reticulum. If these proteins are made in the rough ER, or the lipids and carbs. So what happens is we're making these materials in the endoplasmic reticulum. Think about it as sort of like the manufacturer. But now, we're not at our final destination just yet. next we're going to the Golgi, which is like this Jabba the Hutt ass looking organelle. I don't know what's going on with the Golgi. It's got the weirdest name, and honestly, it's like the funkiest looking thing I've ever seen. But what is the Golgi in charge of? It receives the proteins and the lipids, and what happens is it receives them at the cyst face. All right, it receives them here, and those proteins will travel through the entire Golgi, kind of like one layer at a time, and eventually exit via secretory vesicle at the end. So they'll kind of travel through the deep of the bowels of the Golgi, going through all the layers, getting modified, and will eventually be shipped out of the trans side, received in the cyst and exited out of the trans. So people always think of the Golgi as the UPS of the cell, which is sort of the basic definition, but it's because it receives and modifies and ships proteins and other molecules. So it receives, modifies, and ships these proteins. And another thing that's interesting about the Golgi is sometimes it receives these enzymes called hydrolases. Yeah, hydrolases. And when it does, it actually, the Golgi makes lysosomes, which are another organelle that's actually part of the endomembrane system. So when the Golgi receives very special enzymes, it'll actually create lysosomes. But before we get to lysosomes, let's take a look at this here, these secretory vesicles. So what happens is once the Golgi modifies the proteins, sometimes they're actually destined to leave the cell. So you can see you've got the precious cargo inside of this vesicle, and it is going to fuse to the plasma membrane and release its contents to the outside of the cell, which is actually the process called exocytosis. So I'm going to go ahead and write that for you. So they fuse to the plasma membrane, cell membrane, and will release the contents into the extracellular matrix, which I've abbreviated as just ECM down here. That's what I'm trying to talk about. So yeah, that whole thing is called exocytosis, which is one of our methods of bulk transport. So sometimes secretory vesicles will go and release their contents out, and sometimes they'll actually bring in various molecules. So I know we got phagocytosis going on here with a little food molecule. And the reason why I'm showing this picture in particular is because I think it does a really good job at connecting our lysosomes and their function to the endomembrane system. So you can see we got the rough ER, the Golgi, and here the rough ER has got those hydrolytic enzymes, and they have gone through the Golgi, and because of those specific enzymes I was telling you about, it actually creates a lysosome. And lysosomes are awesome because they fuse to these other vesicles. So here we brought in a little tasty green, whatever that is, okay, bringing that in, awesome. And this is a little food vacuole, so we brought in the food molecule. And I mean, the cell can't, just like you can't really digest things without a stomach, the cell's not going to be able to digest anything without a lysosome. So lysosomes sometimes are called the stomach of the cell, and that's because their job, just like our stomachs, are breaking things down. And the reason why these are really, really important is because they provide a safe place to have a lower, more acidic pH. So you can't just have pockets of pH just randomly floating around, digesting whatever they want, right? There's got to be order to the chaos on a cellular level. And the lysosome aids in that. So lysosomes can break down food molecules, they can break down old organelles that are sort of past their prime and need to be scrapped for their parts. It's a really, really dark world in the cell there. but you know, it's the circle of life, I guess. So the lysosome has a lower pH. I'm going to write that. And just provides that safe place to digest larger molecules into smaller ones that could be used. They have that lower pH. So again, I wanted to reiterate down here that this, sometimes I feel like it gets confusing. You know, you don't really see how lysosomes fit into the puzzle that is the endomembrane system. But remember, these are created with hydrolytic enzymes that are synthesized in the rough ER, and they're sent to the Golgi, and the Golgi actually forms the lysosomes. And lysosomes also can fuse to secretory vesicles and break down the contents inside those. So that's how it can also connect. And then again, that they're going to be used digesting food and things like that. And I want to point out that this process is endocytosis, just making a connection. That's covered in a different video as well. So that's really the gist on the lysosome stomach of the cell. And lastly, I'm going to try to include some of these questions on slides. The whole point of this is I'm going to put the answers in the description. So you would get the most out of these questions if you try to answer them on your own first as a review, and then check your answers using my explanation in the description. So the first one is, what is the path that a secretory protein takes start to finish, aka like what organelles is it rolling through, right? Where is it starting? Where is it winding up? Liver cells tend to have a higher concentration of smooth ER. Why does this make sense in terms of function? So again, the whole connection between structure and function is huge. You must understand how those things are related. And then a hint for our test we'll have in class, that if you see a diagram of an organelle, you really should be able to identify what it is and then also be able to describe its function. So give these questions a try and then check your answers using my explanation in the description.